Hello and a very warm welcome to this Cell and Gene Therapy Insights Expert Roundtable with Miltenyi Biotec. I'm Charlotte Barker, an editor at BioInsights, and our topic for today is going to be smooth cell therapy analytical assay translation from analytical development to QC. We're going to be talking about challenges and strategies for assay translation in the cell therapy space, with a particular focus on flow cytometry. We're joined today by 4 expert panelists to help shed some light on this critical topic. First up, we have Hadar Adams, who's Director of Analytical Development at Atara Biotherapeutics, an allogeneic T-cell immunotherapy company. She has over 10 years of experience in development, optimization, troubleshooting, qualification and validation, and transfer of analytical methods. Ilya Shestopalov is Vice President of Analytical Development and also the Analytical Product Lead at Bluebird Bio. His research focuses on the development of cell-based assays for hematopoietic stem or progenitor products, CAR-T products, and lentiviral vectors. Kitman Yang is MSAT Analytical Manager at Miltenyi Biotec, with 15 years of experience in cell and gene therapies. Her expertise in tech transfer, analytical development, and quality control testing supports the production of Phase 1 and 2 GMP cell and gene therapies. Takale Teklemariam is Associate Director of Analytical Development and QC Assay Transfer at Miltenyi Biotec, where he leads QC assay development and transfer, assay qualification and validation, stability studies, and QC in-process and release testing in cell and gene therapies for multiple clients. Welcome everyone, I'm looking forward to a great discussion today. So let's start with a bit of scene setting question. What are the unique challenges then of developing quality assays, in particular flow cytometry assays for a cellular therapy? Ilya, do you want to go first?
Yeah, I'll tackle this one. So obviously flow cytometry assays, they're integral to cell therapy. It's just like mass spec is to biologics or NMRs to small molecules. They're very key. And I think having been doing this for over a decade, in my opinion, a lot of the historic challenges in developing flow cytometry assays have been resolved in the last 10 years. So we used to struggle with a lot variability in reagents, the difficulty with instruments, the lack of standardization. But I think those are no longer challenges. Same with software. There's a lot of 21 CFR Part 11 compliant options out there. So I kind of thought there's still 3 areas where there are struggles. One is a strategic challenge in how your assays are designed. So, for example, a lot of companies design what's called IPV panels. So you're trying to measure your identity, purity, and viability in a single flow cytometry panel, maybe 4 to 5 colors. That's fantastic for IND and very efficient, but that may cause lifecycle management issues later on. So, for example, what if you want to switch to a different viability modality? And now your validated panel has viability markers, might not be so useful. Or what if one of the reagents that you validate your panel with becomes unavailable? Now your whole panel and the whole assay goes offline. So I think one of the challenges is strategic. You need to decide early on, for product release, do you go for one multicolor flow panel, or do you break it up into smaller panels, less compensation issues, perhaps less problems that you can encounter, and easier lifecycle management. And that could be hard to align on strategically within companies. The second area is positive assay controls. So negative assay controls, super easy. You got isotypes, you got FMOs, you can see where to draw your gates. But how do you know your instrument's actually performing properly? And so oftentimes you need to have a positive control, some sort of mimic or cell or something that has fluorescence that's equivalent to the population you're trying to measure. And that could be quite non-trivial sometimes. There are great reagents out there for some cell types, for others, not so much. And then the third area I think folks need to think about is assay transfer when you're doing manufacturing site transfer. So in a lot of cell therapies, flow cytometry is done with fresh samples during manufacturing at the QC lab within your manufacturing site. Later on, you might want to open another manufacturing site or switch sites. And then once you manufacture products at those new sites, when it comes time for a BLA or MAA, you're going to need to demonstrate comparability of your products across your manufacturing sites. And to demonstrate comparability, you have to first demonstrate assay equivalence. And the gold standard is testing the same sample at your old QC lab and the new QC lab. But if you've already closed your QC lab as you moved, that's not possible. Or if you're working with fresh material, that becomes not possible if you missed that opportunity to do that during site transfer. So, so then, your comparability can become quite tricky if you no longer have the old QC site to test to do your site-to-site equivalents. So I think those are kind of three challenges. They're more strategic, in my opinion, or two of them are more strategic, in my opinion.
So I agree with Elliot that I think that the key challenge, in assay transfer definitely is the control. So in our case, a lot of, because we are CDMO business, so we have a lot of clients sending us their R&D stage assay. So when the assay come in, a lot of time they don't have sample acceptance criteria. And in some unique cases, they don't even have system suitability set up. And some cases that they may have a positive control, but not very robust, and they don't have a negative control. So having like a control, a very robust control, and have a statistic relevant data to back it up is the most important thing. And a lot of time we also encounter issue is that to the client have no ATP in place, which is like analytical target profile, meaning that they don't identify all the critical reagent. They have no data to back it up. They have no other, like Ilya say, that if the reagent run out, they have no bridging study, basically any backup antibodies.
I think that's the point both speakers touched. I know one of the challenge is, especially in the CDMO business, is how do you, balance, instrument availability? Because right now there are out there different kind of instrument, from different companies, and it really to think about it is, especially on the release assay, you have to make it as simple as possible. As Ilya said, companies come up with a fluorochrome of 8 or even more than that. And in that case, it becomes very difficult if you have, I know the current instrument compensate well, but never been compensation is very perfect. And there are always some overlap. So in that case, minimize the number of fluorochrome and focus on a target that is very critical is a very, very, very important. Yes, positive controls. There are no, for CAR therapy especially, there are no standard commercial available. So you have to really establish your standard from the very beginning. Make a cell bank that works well. And characterize, and that can be used, for any kind of to demonstrate your comparability from site to site. You don't have to go through all the manufacturing process to show comparability. You can actually use a well-characterized positive control. The point is very, very to having a well-established method from the very beginning.
One thing I can add is at the beginning of development, one of the challenges can be material. So usually the process development and the analytical development are happening side by side. And sometimes it's hard to get representative samples. The process is continuously changing, so the samples are changing. And that can be a challenge for the assay development. So working closely with the process development team, understanding the changes, how impactful they are on the final sample, risk analysis for the changes. I think that's also important in the development side of things.
Absolutely. Okay, so what then are the key challenges or maybe the most common mistakes that you see in translating assays from analytical development to QC teams? Kitman.
I think the most common mistake, as I mentioned earlier, is that when the assay translated to QC, and then frequently they actually lack the foundations of like a statistically relevant data. and that compromising a robust analytical procedures. So that, as Heather mentioned, that at the early development phase, that a lot of material is not representative. So it's really hard to accumulating that statistic relevant data. But that data is actually later on will be very important to setting like system suitability, acceptance criteria, or even when you enter as a qualification validation, those data is actually particularly important. And that's the common mistake. How to solve the common mistake is basically have the ATP in place as early on during development, exercise the gap assessment. So when the assay coming in through analytical development and through like R&D assay, translate it into assay development group. So put them like basically nail down all the ATP analytical target profile, and that's another most important thing. And as a CDMO, and we always deal with like a rapid timeline. And we need that like the client to understand that sometimes to deal with IND filing, less is more, because they keep thinking that if we have all the assay as a release assay, the chances that they will get a successful IND filing will be higher. But in reality, a lot of assay is not necessary to be a release assay. We can put it as a categorization assay, for example, potency assay, right? If the platform is not robust enough, and we should put it in like as a categorization assay instead of like putting it so early on as a release assay, because later on when we entering like phase 2, 3, and it's very hard to change and justify with the FDA why are you changing the assay platform. So that is like the most common mistake.
I'll just add some really simple things that should be checked off the list. Different equipment, configuration of the instruments, workflow capabilities are very different between analytical development and a QC lab. There's a lot more things in place in the QC lab, a lot more boxes to check off. Sometimes there are reagent requirements. So for example, in an AD lab, you might be able to use an RUO antibody, but maybe the QC lab doesn't permit that. So those kinds of things need to be discussed in advance of the transfer so that on the AD side, they can make sure that they are developing the right kind of assay that can easily transfer into the QC lab. Another thing related to that is reagent traceability. So we've talked about controls and system suitability. Those are going to come from the AD lab. Again, the documentation and traceability within the two settings can be very different. And so the QC lab might not accept reagents that the AD lab, routinely uses if there's not enough traceability and documentation and characterization of those reagents.
I'm glad you brought up instruments. The other piece that often happens is, let's say you have two same instruments, and AD basically provides the assay, the analytical target profile, and they provide a gating strategy. So I think, AD would provide a template. Like, here's the template, load it up on your instrument, stuff goes. I think that's a mistake. You need to have not just a template, but also a gating strategy that's really well described in the SOP. So for each gate, here's what you should see, here's representative examples, here's, sometimes you'll see these funky things. And if you do this, there should be some flexibility to adjust parts of the gate. That is acceptable. It's, that's part of your method. And that's, as long as that's how the procedure is done during validation, that's acceptable. It's not a rigid template. But just throwing a template at the QC lab is a mistake.
One point to add is particularly, yes, documentation is one has been raised. In a QC setup, sometimes let's say if you're actually staining your cells for 20 minutes, you put 20 minutes. Actually, in a QC setup, there's no 20 minutes. There has to be a range. So this is a lack of study in a whole time, for instance, robustness study. How do I know that is my cells are actually behaving correctly if I add 7-AD for 20 minutes or 20 minutes plus or minus 50 minutes? This is a critical step. Always me as a receiving unit from AD, sometimes they give, for 20 minutes, but my GMP partners say, well, if auditors comes, there's no way I can stop my time or 20 minutes. There should be some way. So I cannot put 20 plus or minus with no any data as well. So those are very simple study to perform during analytical development, but it's not captured anywhere. So I think robustness in terms of the whole time, in terms of sometimes the number of cells, in terms of the instrument, you have to, show that so that there's no variability. I think robustness whole time is a critical aspect when it comes to the QC from AD to QC.
What practical steps can teams take to minimize risk in assay translation? Hadar.
So this was brought up just now. I think the number one thing a step that teams can take is to have very well characterized assays. So as was mentioned, robustness, ranges for incubation times, really understanding the requirements of the reagents. I mean, it's really little things that can become really problematic when you're transferring. So really understanding when you thaw a reagent, are you thawing it at room temperature? Are you thawing it on ice? Does that make a difference? How long before you have to use it? All those small steps can make a big difference when you're transferring an assay from one lab to another, because they often get overlooked. So I think that's the most important, and understanding what are the very most critical steps. In some steps, you can pipette differently. Maybe other steps you have to pipette in a very specific way. Not so much in flow assays too, you want to minimize bubbles, let's say, at the end. So maybe you do a reverse pipetting. If that's an important step, it needs to be outlined. It needs to be highlighted in the SOP during training. So I think that's the number one. Another key step is gap analysis. So at the beginning of the transfer process, you have a conversation between the teams. You look at every step of the procedure. You look at all of the instruments, all of the reagents, and you understand the capabilities of the different labs and see if there's any key differences. If there are, then you need to look more deeply into those, understand, is this something that can be worked around or not, and proceed accordingly. So I think that's really important. And along those same lines is also staffing and training and skills of your staff. So we talked a little bit about gating, and that can be really tricky. And if you don't have a very skilled person in the QC lab, then you need to provide a lot more information in the SOP about gating. I mean, it should be there anyway, of course, but you can run into problems if you have this incompatibility of the training in the different labs. So I think we'll talk some more about communication. I think that's really, really important. So in all of these steps, for the QC lab to be aware of all of the development, all of the robustness work that the AD lab has done, being able to communicate freely and often is really important, because again, it's small details that can trip you up later. So being able to communicate effectively is important. And finally, I would say hands-on training. This is really, really critical as well. Being in the lab, being in the transferring lab, seeing how things are performed, the workflow, even the orientation of equipment can be important, and kind of seeing how that might be different in the QC lab, and are there going to be any challenges? We had a situation where we had two very similar cell counters. Same technology, same manufacturer, but one was high throughput and one was not high throughput. And that ended up being really critical because there were so many cell counts. And it could add a significant amount of time if you have to do them one at a time versus in a high throughput situation. So really understanding that, and that came out during training. So training in the transferring lab and having that hands-on training, I think, is really critical.
definitely the points are very well taken, particularly training and the follow-up are very critical. The reason being, usually assay development in the AD is performed particularly using healthy donor. And when it comes to the QC, mostly they are in the patient samples, and which sometimes behave differently. So the sometimes the plots, maybe the analysis plot put in the SOP might not typical of those the patient sample. So the training and the follow-up communication between the two groups is very critical. That really somehow makes the interpretation of data differently than what is being developed during the AD. So yes, training and communication is a critical part of. I think that's the only thing I can add on this one. Yeah, it's already been mentioned.
So to follow up that different instrument, even though it's like almost exactly the same thing, I think that Miltenyi, we have like the MaxQuant system that we have the smart gain functions that if we transferring assay from a different site to site, I think we can use the smart gain function that because we can basically align with including the instrument setting. So not only just dump the template, analysis template from AD to a QC, and then actually using express mode and the smart gain function is actually can minimize all those problem a lot. We still need to have like an analytical team, like a ASAT team, MSAT analytics, to basically draft like all the details on the gating from healthy donor to the patients. But I think that with that, like the Miltenyi technology, I think that would definitely help out a lot.
So I think it's fair to say then that communication emerged there as a key theme. Can you suggest strategies for effective communication between teams during assay translation? Kipman.
The first thing is we all have to have like a collaborative mindset, including the client as a CDMO business. So we talk about, we go through like very early on a gap assessment. We basically have like a paperwork exercise and a PSD in place, a very detailed, comprehensive PSD in place, so that we can have all the, we basically on the paper, we'll work through all the details to see what is needed and what is a must-have, what is like a good to have. So with that communication in place, with that collaborations mindset, that's number one thing is very important. The second thing, I would lean heavily on the PM, actually, the project manager. So because we taking care of many client projects, and then so we need a project manager that, well, first of all, like you have to be technically, good enough that is able to take a very good meeting minute. And because sometimes, when we're talking about like the Prodigy, the CAP, and all the other system, it sounds like a foreign language to a PM if they if they don't have like a cell and gene therapy background or even a science background. So a PM that is actually orchestra all the meeting and understand who's the key member definitely need to be come in a certain meeting and make decisions. So it often happen is the PM schedule one team member come into the meeting and try to make a decision, but the decision is impacting QC a lot, but there's no representative to representing QC. That actually happen a lot of times. Yeah.
I agree with what Kipman said. Being able to have a say in the transfer. AD has to have a say in that transfer. I also, I think as the originating lab, once the assay is within QC, it shouldn't be treated as, okay, I threw it over the fence, it's over there, not my problem. Ability to, for example, be part of the assay trending. AD probably has more experience with this assay, so they should see the trending data, what controls are being done, how is that going, what are your trend charts. When things go wrong, AD should be able to have access to raw data, FCS files. There's no reason why QC lab cannot share the raw data upon ask to AD. Ideally, in an ideal situation, you go as far as allowing AD to have a part in training new analysts in QC, not just during the first assay transfer, but as new analysts are brought into the assay, can they come to the AD lab and hang out with us for a little bit and see the assay being run over here, so then they can come back to QC and be like, well, this is what I saw over there, and that helps me visualize, or another approach is AD can provide training videos. Sometimes an SOP only goes so far, but if you just record someone doing that procedure and provide it to QC, that could be really effective for assay transfer.
Yeah, I agree what Kidman and Lai said. Definitely the AD team communication across all stakeholders is very critical. I know sometimes once the assays transferred into QC, there are unforeseen outcomes such as invalid assay always, all those things. They definitely the SME who have expertise from the AD is very, very, very much critical, and the understanding help. It doesn't mean the QC team is not knowing what they're doing, but from a technical perspective, it makes sense to have from the originator as a backup information is very, very critical. So yes, communication, even the assay goes to the next level. We don't know. Assays are assays. Sometimes really needs improvement in all the way into the later phases. So definitely communication and help from the originator, particularly the AD team, is critical during our clinical lab testing.
I'll just add that I think face-to-face communication is critical. Things get lost in emails, things get misinterpreted in emails, so I would not rely heavily on emails. I mean, obviously, that's a way that we communicate, but I think having frequent face-to-face, even just short, frequent check-ins for the QC lab and the AD lab to get questions answered and check on minor things, I think that's much better handled in a face-to-face format. I've had lots of situations where things are just slightly misinterpreted, and it can make a big difference. I think we talked about this before, but just prior to the initiating the transfer, I think having all the documentation handed over from AD to QC so that they can review all of the development work, the robustness, the procedure very thoroughly before you start having this conversation, so that they can be as informed as possible and can be ready to ask questions and dig deep into the procedure. And I think it's really important to have the analysts as part of this conversation, not just managers and supervisors. We need the people that are in the lab holding the pipettes. Those are the people that need to be part of the conversation.
Absolutely. What would you say is the impact of running QC internally versus externally? What's the impact on communication there, would you say? Ilya?
It's a great question. A lot of companies struggle with this because setting up internal QC is very expensive. So just some definitions first. So when I think of internal QC, I think internal to the company. So your Bluebird, this would be Bluebird's QC. And if you're working with an external CMO that has their own QC, to me, that's still external QC because it's over there at the external CMO. And personally, I've seen both models, and I've seen them work and I've seen them not work. So I've seen where you have an internal QC lab that's really hard to work with, and I've seen external QC labs that are great partners. What often happens, whether even with internal QC, is they tend to silo themselves away, and there's a reason why they do it. They do it to manage the risk of GMP noncompliance. They don't want AD walking in willy-nilly, watching over their shoulder. They try to be kind of in a silo. There's a reason for that. But at the same time, as we were talking earlier, we also want the QC to be inclusive, because you need to have effective communication. If that silo is too hard of a firewall, you can't effectively have the face-to-face meetings that Hadar has brought up. you can't have the proper training. So I think one of the differentiating factors, in my opinion, whether QC, internal or external, works or not effectively, is what is their mission. If the mission, if they're somehow vested in the success of your product, that they're going to behave differently in terms of how they communicate with you. Whereas, and it's very different from if they're just getting paid a contract to test your samples. It just comes across very different. So you have to work hard to establish that mission-driven approach, whether your QC is external or internal.
I agree with all the other speakers mentioned. So like being very inclusive, and especially even very early on, at Miltenyi, we try to including our QC team even in the PSD and now in the ATP planning as well, analytical target profile planning, because they are the one it is in the front line in the war zone, like holding the pipette, so they should know what assay is gonna come to their lab, and all the idea that it should be communicate with them very early on, so they can feel like they are like inclusive, and then not feeling like, oh, if that's assay OOS, it's always blame on, it's the man issue. But sometimes it's not, right? And then we all have to have that open mindset that, hey, let's take a look. Is it really the man, or it could be the assay? It could be, is that something that we all missed it during the ATP? We did not evaluating. That actually is a critical factor to impacting the assay result.
Yeah, I think Ilya brought up a really good point that it's not always internal is better, external is harder. I think it's important to understand the differences between the AD setting and the QC setting. It might be a little bit easier if it's an internal QC to get that information, how their processes work, what workflows do they have, what systems do they have in place that we need to accommodate in the procedure. So that might be a little bit easier if it's an internal, but not always. So I think it's important to understand that. I think also one of the things that can be challenging, I think more challenging when it's an external QC, is coordinating the on-site training. So a lot of times this has to be, thought of really early in the agreements between the two companies, how many on-site visits are permitted and that kind of thing. So it's important to include the AD and the QC personnel in those early conversations when you're drafting the SOWs about how the companies are going to work together. Because as we've mentioned before, the hands-on training is really important. So that can be just something that needs to be, highlighted if you're working with an external QC partner.
Just one point I will add. I know Miltenyi in San Jose currently almost most of the analytical methods are in-house QC testing, but we do have some safety tests that are done in external lab. And one of the things which we do actually with the external lab, there are some bi-weekly meeting to follow up how is the progress. For instance, if you send samples for lab release, we have to really communicate with them and then have a follow-up meeting so that they can really get prepared to test the sample on time. So this meeting is really very critical, whether it's done between QC internal, external, or the SMA of the internal with the external. So please make sure that you're having not only sending emails, some emails can be missed as previously stated. Follow-up meeting, bi-weekly is typically we do that. Even if we don't have any topic of discussion, we'll ask any challenges. We'll be sending this amount of samples now, next week. Do you have the resources? Do you test, for instance, on the weekends? All those sort of activities. So, a follow-up meeting is very critical when you are really, relying on external QC testing.
And may I add something, Kelly, to that? So you mentioned you send samples. I think also what's really, really important is to have like a common SharePoint set up, so that those external QC labs, even if it's preliminary results, just pop their raw data right in there. So we don't have to every time email, hey, how's that sample going? Hey, how's this going? It just cuts down on the noise. We can just go in, see what's on SharePoint, and there's sort of this agreement that They're not going to just randomly delay. If they've actually run it, it's going to appear in SharePoint way before we get like a certificate of testing. So that is a fantastic communication strategy when you're not at the same site.
Which technological advances can smooth the transition from assay development to QC now and maybe looking ahead to the future as well? Hadar.
When we're talking about flow cytometry assays, I think the biggest challenge is the gating. So in terms of automation, the flow cytometer is very automated, but the gating can be very manual. So I know there's been a lot of work being done on this, which is fantastic. And this kind of goes back to like one of the first things we talked about was having simple assays. So if you have a very simple assay, then the gating will be simpler. If you have so many different colors and populations, obviously things can get more complicated. And then automated gating is not really an option. But I think in terms of the variability that we see in flow assays, and how can we minimize that, I think automation is one of the key, automated gating. I think another thing is having really good controls. I mean, reference standards are not always available, but having good positive controls, it might not help in the transfer, but it really will help in the troubleshooting. You know, if there's an issue in the QC lab, helping pinpoint like where that's coming from, where that variability, where that issue is coming from.
I totally agree with Hadar. Being in a QC, being in the lab as well, definitely, automatic express mode gating is one of the important factor. However, when clients are actually coming from AD, they don't, see the benefit actually in this because they used to, manual gating, adjusting. In addition to making the assay simpler for the analysis, this is also actually very good for regulatory. It has a regulatory impact because you don't have to really. How do I know? I put the right population on the gating, and sometimes it just flow cytometry. There are sometimes subjectivity, which is obvious. Although the result might not vary much, between analysts, which can be demonstrated during assay qualification or validation. From the point of view of getting the result very quick for manufacturing process to continue. Sometimes the result could be intermediate product that is needed for the process to continue. In that case, automation is a very critical. Yes, that is true as well. The Max Quant has in the automation system, especially the express mode. It can be adapted for different kind of assays. It's not for one. And in such cases, then if you have a car, let's say, if you're measuring, for instance, the transduction efficiency, we typically most use, the bright, color. In that case, it can be automated as a platform for different clients. And definitely, automation like analysis is the big challenge. And so in that case, express mode is very critical, which we have it in here and really solve the time as well as from the regulatory perspective, you just generate one data, everything is there. So yes, a good point, definitely.
First of all, on the automated gating, I agree. It has a lot of potential. The danger is that once you validate, let's say you have an express mode version 3, you validate your assay, and then a year later, the vendor comes out with express mode version 4, and that's they kind of force you to move to it. Well, you're going to have to revalidate the assay. So if automated you have to be able to, if you're going to use that approach, it's a great approach, but you need to have an understanding with the provider of that approach that you will be able to continue using that automated module without any updates in perpetuity. You cannot have a new version come out that auto gates slightly differently. So that's the challenge in a technology that's constantly evolving. From the other technological piece that I find really intriguing are these microfluidic based flow cytometry systems where it also automates the staining. All the reagents are already in there. You just literally throw your cells in. And, it parallels a lot to how cell counting has evolved, where even 5 years ago, pretty much everyone was doing some sort of manual cell count, trypan blue. And now everyone's using, the industry standard has become these single-use chips that you literally pipette your cells into, everything's fluorescently stained, and is much, it's really the industry standard for cell counting. And I think flow cytometry is going to go that way too, once the technology matures to accommodate the slightly more complex gating than just viability. But again, with any evolving technology, you need to be able to have a lockdown version that exists in perpetuity, or it's going to screw you over in your lifecycle management.
So I think standardization is like one of the way that can actually transfer the assay from AD to QC more smoothly. At Miltenyi, we have like a standard express mode. We even have like reagent that is in one vial with all the cocktail is a dry down form. And so during the analytical development, so we can just use that was kind of like Sting Express kit, and then been using and collecting a lot of data. And then for QC, and then so we have a lot of data already, and using a standardization express mode and a standardization gating strategy. So that actually will help actually collecting a lot of like data as well. And the other thing will be automations. So we have something that, in the pipeline, but it's not ready to share with our client. But at Miltenyi, we actually developing some like robotic system. So it will be very standardizing the workflow.
Just to finish up, I wanted to talk briefly about the regulatory considerations. What would you say are some of the key regulatory considerations in validation, qualification, regional differences? Takeli, do you want to start?
I think I know the term assay validation and qualification sometimes is being used interchangeably. Sometimes it's just not able to know. But in general, both assay qualification and assay validation are being performed to check the fit for purpose of any method down the line. And typically the ICH guideline, ICH Q2R1 is the guideline that both assay qualification and assay validation are used. However, there are some differences between assay qualification and validation, particularly in terms of the depth of the study, as well as the phase where assay qualification and validation are done. In the assay qualification, I know the attributes that are being evaluated are almost similar to assay validation. However, during assay qualification, early stage in the development, during the phase 1 are being performed. And typically during assay qualification, a single representative lot can be evaluated to demonstrate fit for purpose of any assay, particularly a release assay. In terms of regulatory requirement, there is no really clear, regulatory requirement to qualify your assay from region to region. There is no clear written guidelines. You have to qualify your assay during the early stage of IND submission. However, it is in the best interest of the sponsor to qualify your assay to demonstrate actually that method is able to be validated in the future. So although the term is sometimes confusing, in the simplest term, qualification is the early stage of demonstrating the method is able to perform as intended in the future, and also it helps to establish acceptance criteria in the future when you validate your assay. So by definition, if anyone find any guidelines, there is no assay qualification guideline, but both validation use the ICH guideline. Typically, they mention what parameters need to be evaluated. Of course, depending on the assay type. However, when it comes to validation, there is a clear requirement from the regulatory agency, particularly in the late phase of IND application, particularly in phase 2 commercial or a BLA submission, and hence, qualifying your assay at the beginning definitely will help to validate your method. And the acceptance criteria typically are similar to the during the qualification, but it can be more tighter. And the other key parameter need to consider during validation, you have to validate not only your final product, but also intermediate controls are very critical part of the assay validation. Although during assay qualification, we don't not necessarily evaluate intermediate product. For instance, if you have a car transduction assay, typically, companies, before they do the harvest, they do actually a day before to check, the transduction efficiency. They use that data for, whether harvest happens or whether you have to extend it. So those intermediate products are actually intermediate assays are very critical. So during validation, you have to show actually, how do I know the method is actually predicting your outcome during the harvest? So validation is required. So in general, from the sponsor point of view, qualifying the method early in the development and even in the phase 1 before submission for IND application is very critical, and someone will be in a best position for the application to be during IND review. So in general, the requirements for validation are very clear, but for qualification there's none. But most all current small companies they do qualify their assay, particularly the release assay. But there is also confusion. But some companies also think every assay needs to be qualified, but that's not the case. There are some characterization assay which you have to really collect that down the lines are very, very critical in your decision making, but you don't have to qualify. You collect those data and just, could be a report result, and then you can qualify when there are multiple, data available. Particularly, patient data are actually critical to, evaluate those kind of qualification methods as well.
I'm not sure if it's a regional difference, but certainly in the US, as you go for the BLA, there's been a lot more scrutiny of your robustness that you might not need early on. So in addition to validation, after you validate your assay, you're already doing your pivotal trial, make sure you're investing in robustness studies, or it might become a post-approval commitment. The other thing I would say is with these assays, typically you're trying to capture some precision during your qualification. That's your acceptance criteria for validation, and your precision might be, really good. But there is a certain temporal aspect to flow cytometry that is not captured by precision. So, maybe during your validation, you saw a 7% CV across, multiple analysts and days. But as you're doing, let's say, stability, which is the same sample now being run over a course of years, you might see that the precision of your assay is actually not as good. So, and you should really factor that into how you set your specs. So, if you're kind of bring an assay online very last minute, and you had maybe 3 assay occasions, you did a whole bunch of historical lot testing and batch, and then you set your spec on that, you're really shooting yourself in the foot, because you really haven't captured that temporal variation in the assay that your validation might not catch. So that's just something to think about from a regulatory point of view. You're going to establish specifications, and how many assay occasions and over what course of time have you done them to establish your specifications.
I think one thing that definitely needs to be discussed early on is when you're talking with a QC partner is where the qualification needs to happen. I mean, I think everybody understands the validation needs to happen in the QC lab, but in the early phases, oftentimes AD does the qualification and then they transfer the method. But there are some QC labs that want to have the qualification performed in their lab. I mean, they're the ones that are going to be performing the assay. So I think that's just something, again, with the communication needs to be understood in advance. Maybe AD does a pre-qualification or does some of it, or I've seen co-qualifications where it's kind of joint between the 2 labs. But that's something that needs to be worked out in advance. And also was mentioned the, characterization assays don't necessarily need to be qualified. I just want to point out the new potency guideline that came out just last month, all about potency assays and how critical the potency assays are. And from my reading, it seemed that the recommendation was that potency assays should be qualified. So it did talk a little bit about characterization versus release, but in the end, I feel like my interpretation was that because potency assays are so critical, you really want to have that assurance that the assay is performing robustly and well. So that's my interpretation.
I think the last point I want to add is actually statistic. So I would like to the magic number, like 3 sample, 3 replicate for qualification. I think that in the future we should have more like a statistic validation. Maybe 1 to a model, maybe not 3 times 3. It's like a magic number anymore. Maybe certain assay, maybe potency assay, maybe like the magic number is 24. 24 reps. Or so all those data is like and very early on try to have a statistic relevant data to back it up, like how many samples we should test it, how many replicate we should have in the assay qualification is actually will help us out, later on when going through validations.
Thank you very much for joining us today for the roundtable, and thank you to our panelists Hadar, Ilya, Kitman, and Takale. We hope you enjoyed the discussion and that you'll join us again soon.